Hello, my name is Sophie Lay, and I will be talking a little bit about blood clot dynamics in Tenrec ecaudatus, also known as the common Tenrec. To give a bit of background, Tenrec ecaudatus are placental mammals native to Madagascar that exhibit plesiomorphic traits and unconventional hibernation patterns. For example, Tenrecs are characterized by ancestral traits such as a smooth brain, internal testes located near the kidneys, and a cloaca, which is a singular opening for the digestive, reproductive, and urinary tracts. Remarkably, Tenrecs have extreme thermoregulatory and metabolic plasticity. For example, Tenrecs can be active or hibernate at ambient temperatures of 12 degrees C or 28 degrees C, and an active Tenrec with a body temperature of 13 degrees C can even swim. And so the spleen is a lymphoid organ that serves as the main depot of red blood cells and plays a role in the immune response by filtering blood, removing old or damaged red blood cells, and clearing microorganisms. In the ground squirrel Spirophilus undulatus, spleen weight, hemoglobin levels, and total and extracted protein content of the spleen increased when the animals entered torpor, reaching maximum values when the body temperature dropped below 25 degrees C. These parameters return to characteristic values upon arousal as red blood cells are released into blood circulation. In Tenrecs, blood cells are also sequestered, oftentimes independent of state or body temperature, displaying a tenfold variation in hemoglobin levels. The spleen engorges with red blood cells and may experience a 13.9-fold variation in body mass adjusted size, meaning that as spleen size increases, blood hemoglobin levels decrease, while splenic hemoglobin levels increase. White blood cells are also sequestered in the spleen, but not synchronously with red blood cells. Here we can see a picture of a small spleen and a large spleen engorged with red blood cells. If we divide spleen mass by body mass and multiply by 100, we obtain the percent mass of the spleen in proportion to the body. For the small spleen on the left, the spleen is 0.135% of the body, but for the large spleen on the right, the spleen is 1.27% of the body. For a quick recap, Tenrec spleens engorge with red blood cells, and spleen masses vary by 13.9-fold. Hematocrit, which is packed red blood cell volume, varies from 17 to 59% in Tenrecs. With such dynamic changes in red blood cell availability, we asked what comprises blood clots. The purpose of this project is to determine the relationship between blood clot formation and metabolic status of Tenrecs by analyzing the relative participation of red blood cells, fibrin, and platelets in blood clots through Western blots. Light microscopy was also used to visualize general clot composition. As for materials and methods, 40 wild-caught Tenrecs were obtained from Mauritius in 2014 and housed in compliance to the Institutional Animal Care and Use Committee approved protocol. The blood clot samples for this project were obtained throughout this summer from animals killed for tissue collection via a primary cervical dislocation followed by pneumothorax. Blood clots were snap frozen in liquid nitrogen, then stored at -80 degrees C prior to preparation for the Lowry assay and Western blot. Blood clots were homogenized in an SCS-containing sample buffer. After total protein quantification with a Lowry assay to allow for constant loading, lysates were electrophoresed and transferred to a PVDF membrane. Standard Western blotting will be performed for fibrin and markers for platelets and red blood cells. Red blood cells, platelets, and fibrinogen will be marked by antibodies for SLC4A1, the band 3 anion transport protein, platelet glycoprotein 1b alpha chain, and fibrinogen respectively. Data for 14 clots, 7 low metabolic rates, and 7 high metabolic rates will be analyzed. For the second part of this project, blood clots were also fixed in 10% neutral buffered formalin for viewing with light microscopy. Prior to embedding in TissueTech OCT following standard practices, the blood clots embedded in OCT were then stored at -80 degrees C, and in order to view single-cell layers while maintaining the integrity of the blood clot, the cryostat was set to -20 degrees C to cut slices approximately 7 micrometers thick. Standard glass slides were treated with poly-L-lysine so the negatively charged slices stuck easily. Multiple consecutive slices were kept on single slides, labeled with the complete animal ID, as well as the order in which they were collected. These slides were then stored at -80 degrees C prior to hematoxylin and eosin staining. For the results, I have provided a few pictures of blood clots all at 1,000 times magnification. In the center, where it says A, there are two different pictures for a blood clot from M18343, which is the ID for the Tenrec. Notice how we could see clumps of red blood cells and single red blood cells. This blood clot also has a prominent amount of white blood cells, which are the bigger purple spots. The reason why there are many white blood cells is that he had an abdominal suture issue that would not heal over the course of 4 to 5 months. For the pictures labeled B, you can basically see lots of red blood cells aggregated together. Also, hematocrit values are all provided. And on the right here are some more pictures of blood clots from F16898, FWC871, and FWC816. All were relatively healthy aside from FWC816, who had a recurring abscess on the snout. Hematocrit values are also shown here. We performed a Lowry assay to determine total protein concentration, and absorbance is read at 750 nanometers on the spectrophotometer. Sample lysates were electrophoresed and transferred to a PVDF membrane, but antibodies have yet to successfully mark the target proteins. But we were able to visualize composition with light microscopy. Next is a graph of a standard Lowry using bovine serum albumin. Using a best fit line for a second-order polynomial, I was able to determine total protein concentration, which is necessary to prepare sample lysates for constant loading in the Western blot. Upon completion of this project, we have a few expectations. In depressed metabolic states, we expect longer clotting time and a higher ratio of fibrin to platelets and red blood cells. In more active metabolic states, we expect faster clotting time and a lower ratio of fibrin to platelets and red blood cells. There are a couple of reasons why it'd be interesting to research blood clot dynamics in Tenrecs. Excessive blood clotting leads to heart attack, stroke, organ damage, and pulmonary embolism. However, further research in the clotting mechanisms of Tenrecs may help us better understand how clot formation is naturally avoided. Thanks especially to the Tenrecs and Dr. Van Brooklyn, who have made this research possible, as well as the National Institute of General Medical Sciences for supporting this project. Thank you so much for listening.